Good day. My name is Francisca Arbulatin, and by this time I'm gonna tackle about some examples of proteins and their structures. Examples of proteins are fibrous proteins and globular proteins, but now I'm gonna talk about the fibrous proteins. So, fibrous proteins are proteins that have a fiber-like shape. Fibrous proteins serve as structure support and tend to be water insoluble. So, examples of fibrous proteins are A-keratin, found in hair, fibrin, found in silk, collagen, found in many tissue cells. So, this is the structure of A-keratin in a helix. So this is the structure of a helix. Coil of two helices, which is combined of a helix, and combined to another helices, it forms a protofilament. Next is microfibril, and next is hair. Our hair is composed of many structures. Even though it is tiny, it has many structures and composites. Fibrin in a B-plated sheet. So this is its structure. So our silk is composed of stacks of fibrin sheets. Next is a collagen. Collagen is a form of triple helix. One stands for blue, one is red, and one is green. Next is globular protein. Have a characteristic compact, rough spherical shape. These proteins are capable of very complex functions and have very complex structures. It is a globular proteins that provides most life machinery. So, some globular proteins are described in the following. Myoglobin. These conjuncted proteins functions as a storage protein, binding oxygen from muscle tissue. Within the center of this structure, it is an organically bound in iron atom in a hemiring. This allows myoglobin to store oxygen. So this is its structure. So hemiring is found in this area. Next is hemoglobin. This protein is a transport protein found in a red blood cell that transport oxygen. This protein has a quaternary structure. It is composed of 4 subunits and an oxygen binding hemigroup in each structure. This is its structure. Okay, so carboxypeptidase A. So this is a catalytic protein, an enzyme that catalyzes proteins digestion in the small intestine. Fatty acid bind protein. This protein is a transport protein which transports fatty acids through the bloodstream. Next is ferrin. This protein is a storage protein in the liver that stores iron. So ferritin is a storage protein in the liver that stores iron. Next is HIV reverse transcriptase. This protein is an enzyme. It is responsible for converting the AIDS virus genetic materials into a form that can infect the host. This enzyme is primary target for drugs designed to block the spread of AIDS. So this is the drugs that can block the spreads of AIDS. So myosin. This protein is a movement protein. It is an important protein in a muscle tissue. So remember, myosin is a movement protein. So next is growth hormone. This protein is regulatory protein responsible for coordinating overall growth. Immunoglobulin G. This protein is a defense protein. Immunoglobulin antibodies as large binding proteins that serves as critical biological function. These proteins bind molecules that are foreign to the body as a defense against disease. So, my immunoglobulin G is a defense protein, which is a defense against diseases. So, what is the difference between fibrous protein and globular protein? Okay, so fibrous protein are long, stranded-like proteins that are insoluble in water, whereas globular proteins have a spherical shape and are soluble in water. Now, let's proceed. Loss of protein's structure. So, the loss of protein structure will affect the protein's function. If a protein loses only its natural three-dimensional conformation, the loss of structure is referred as denaturation. So, denaturation involves the alteration or disruption of the secondary, tertiary, and quaternary, but not primary structure of protein. So, denaturation may involve irreversible alteration caused by a slight shift in pH. To the extreme alteration involves the tanning a skin to form a leather. So, another example of this denaturation is when you cook an egg, and when you break it, you can see the egg whites, and when you turn on the heat, the egg whites were turned into a white because the egg whites is colorless, and it turns to a white color. So that's one of the example of denaturation. So structural change occur when a protein molecule is denatured. During denaturation, the hydrogen electrostatic and disulfide bonds are broken, resulting in a change of structure and properties. This figure below shows what happened to a protein molecule during protein denaturation phase. So this is example in when you want to be a tan, your skin want to be a tan, and you be directed to the sun, your skin will turn into a change in color. This is the example of denatured protein. So, loss of protein structure also occur when a hydrolysis of peptide bond to produce free amino acid. This chemical reaction destroys the protein's primary structure. Proteins can be hydrolyzed by boiling in a solution containing a strong acid such as HCl or a strong base such as NaOH. So this is its structure. Peptide linkage in protein chain and the rest of the protein chain. And that's all. Thank you.